Perfect. So let's introduce Professor Hari, which is our first, the first presenter in this kind of seminar. And I have to say that first I invited him. He was supposed to be in May in Brazil. So he was supposed to come in Bonito for the SBF autumn meeting. So normally he was supposed to go in the beautiful place of Brazil, also pass here at the Institute to work and to collaborate and to give some more talk. But actually, so he was not able, he was not able to come to Brazil due to the, of course, the situation. But we decided to invite him as the first of the first seminar. And we are still planning when it will be possible that he will continue, he will be able to come to visit us in Brazil. So Professor Hari is a professor at the University of South Florida. He's part of the Department of Physics since the year of 2000, where he established his lab is the Functional Materials Laboratory. But basically is working on everything that is nanomagnetism. But it's a little more ample. You will see that he is working in several topics at the same time. And I need to say that just now, he turned out to be a distinguished university professor at the University of South Florida. So I have to say congratulations. It's a good news around all this news about the pandemic. And he's also a fellow of the American Physical Society, APS, specifically for this contribution to nanomagnetism. But nanomagnetism for him range from magnetization dynamics, also everything that is refrigeration, magnetic refrigeration, spin calorimetrics, complex oxide, and nanoparticle. And the topic of today will be about nanoparticle, specifically for nanomedicine. So it's one topic among all the topic that is working on. And well, I just need to say also that he has over 265 publications and a lot of invited talk around the world. And especially, he's part of IEEE. And is a senior member of IEEE. And last year, when it was possible to travel around the world and so on, he was appointed as IEEE Distinguished Lecturer. So for all the year, he was going from one place to the other. And actually, basically, the talk that he will offer us today is the talk that he was giving last year about magnetic nanoparticle for medicine application. And so I will not take more time for Hari. So thank you very much to be with us today. I hope it will be the first of many other times that we will be able to be together online or physically sharing something together. So thank you very much.
Thank you, Fanny. Thank you. It's now my turn to speak some Brazilian, so I did some practicing. Olá, boa tarde, caros alunos e professores. Meu nome é Hari e sou da Flórida. Obrigado pelo seu gentil convite. Por favor, mantenha-se saudável e seguro e venceremos o vírus. Falo português graças ao tio Google. So if I make any mistake in my Portuguese, whether you see them on the slides or when I speak, please blame Uncle Google, okay? I use Google Translate to make some of the words. So thank you very much. It's a pleasure to be here online with you guys at Campinas. I was actually 3 years back, I visited the Federal University in Rio de Janeiro. I was there for a week as a visiting professor and gave a series of lectures. So I've been to Rio, and that's the only city that I've visited in Brazil. So I'm looking forward to coming to Sao Paulo and Campinas and hopefully Bonito at some point in the future. So thank you guys. Thank you for showing up. I can see that there are quite a few of you, more than 50. Wow, that's a great audience. So it gives me a lot of pleasure to tell you a little story. So today I'm here to tell you the story of anisotropy, as it says on my slide in Portuguese, papel de anisotropia. So the whole idea is that what is anisotropy? Anisotropy, of course, in a very simple way, is that properties are not the same in different directions, right? So that's anisotropy. So what I'm going to do is talk about specifically how anisotropy can be manipulated, can be engineered in nanoparticles, and how one can use that for nanomedicine, especially for therapy and diagnostics. And there's a new term. Even nanomedicine is a fairly new term, right? It's been there for maybe 20 years or so. And theranostics means therapy and diagnostics. So it turns out that magnetic nanoparticles can be used for both diagnostics of cancer, as well as therapy, as well as treating cancer. So that's my goal today, to try to present to you some results, mainly from my group, but as well as give you some idea about the work going on in this field. Now, I'm going to try to speak slowly. I know that English is not the native language for many of you, although I'm sure many of you doing PhD and in the university system are quite comfortable with English. But I'll try to make sure that I speak slowly so that I can make some sense. I also have some of the slides, some of the words and some of the sentences I tried to, again, translate to Portuguese for your benefit. Okay. All right. So first of all, let me say that I would like to thank you again for welcoming me. And I would like to certainly acknowledge all these agencies that have funded our work over the years. Mainly, most of the funding for us right now in magnetic materials comes from the US Department of Energy. Now, you may be already familiar with the National Science Foundation, and I'm sure there are very similar agencies in Brazil to this. It turns out that a lot of material science work is funded by DOE, Department of Energy. So that's a big source for many of us. In fact, most of the solid state condensed matter physics work is funded by DOE. A bigger amount is funded by DOE than the NSF, which is something that not many people know. Okay, so again, thank you very much for inviting me to IFGW. And if I go to the next slide. Well, it's not going here, so let me make sure that, can you see me? Can you hear me?
Yeah, we can see and hear you.
But I'm not able to control the slide here. So what I'm going to do is that I'm going to, for a brief 10 seconds, I'm going to stop sharing, go to the PowerPoint, pull it up, and then see if I can do it again, okay?
No problem, actually. Perfect. So I was doing some instruction for.
Yeah, I'm just going to share. I think I was sharing a difference. Now you should be able to see it. Can you see it now?
We can see Florida, yeah.
Yeah, there you go. That's what I wanted to show you. All right, so I wanted to show you where I come from, where I am right now sitting. And if you look at the Florida coast, this is Florida, of course, water all around. And I'm here in the city of Tampa. This is on the west coast, or the Gulf Coast of Florida. The beaches here are some of the best in the country. So this is not just me saying about my city. It turns out that there's a magazine that ranks all the best beaches in the United States. And Clearwater Beach. Clearwater is actually a neighborhood of Tampa, and Clearwater is always #1. Okay, so we have beautiful sand beaches. And it's a great place. You know, right now it's really sunny and nice outside. It's summer here. Summer is beginning, so it's starting to get a little warm here. Okay, so a few words about my university. I would like to welcome you all to Tampa and also USF, University of South Florida. It turns out that we are a pretty large public university in the country. We are the 12th biggest in size. We have about 47,000 students. About 35,000 are here, right here in Tampa, which is the main campus, and the remaining students in other branch campuses. And in the Department of Physics, we have something very unique. We actually offer a PhD in applied physics, and this is the one of its kind in the state of Florida. You may have heard of other big universities in Florida, for example, University of Florida in Gainesville, and then Florida State University in Tallahassee, where all the big magnet labs are. Magnet lab is, the large magnets are. That's in Tallahassee. However, it turns out that they don't offer a PhD in applied physics, only we do at USF. So if you want to get a PhD in applied physics and live close to the world's best beaches, so that's, we are the only choice. So please pass the word around. If you'd like to, students, if you'd like to know more about the department, you can please visit the website or email me for any information about the kind of programs that we have. Materials science, condensed matter physics is the biggest research groups that we have. They are the biggest research groups in the department, both experiment and theory. So again, we have a lot of work going in many different areas. Magnetism is only a part of it. There are 3 of us working in magnetism, and we have a surface physics group. We have lasers, group working on lasers. We have a group working on biophysics. You know, many kinds of materials. Okay, so I would also like to start off by acknowledging my entire group. These are some of the people right now. Some of them have actually graduated, but I put their names here because they contributed to this work that I'm going to talk about, which is magnetic nanoparticles in nanomedicine. These are some of the postdocs. Dr. Raja Das is right now an assistant professor in Vietnam. He is a key player, and some of the work that I'm going to talk about in nanotubes, in magnetite nanotubes, they were actually made by him. And another important person here is Dr. Javi Alonso. He's right now an assistant professor in Santander in Spain, University of Cantabria. And again, he was instrumental when he was visiting postdoc in my group to set up a lot of work in trying to look at the effective properties of these magnetic materials, magnetic nanoparticles for treating cancer. And here are some of the students that worked in this. The ones in green are the ones that really took part in the specific research that I'm going to talk about. And as Fanny mentioned, we work on a number of areas, like typical groups here. We have to be active in different areas because of the large number of students. And essentially, we work on a large number of areas. And if you're interested, you can always talk to me later. But today's topic is going to be on magnetic nanoparticles, specifically applied to nanomedicine, and also specifically for cancer therapy. So that's basically what I have in mind. Our lab has a lot of facilities for making materials and measuring all kinds of magnetic properties. And here are some of the experiments that we are currently doing. We can measure the magnetic properties using a squid magnetometer, a PPMS. These are all instruments that are used, magnetometers that are used. I'm sure there are some in your campus too for measuring the magnetic properties. We can do transport measurements. There are also 2 experiments that are very specific that is somewhat unique to us. And I'm going to talk about both of them because they're relevant to today's talk. One of them is transverse susceptibility. We actually can measure anisotropy precisely, very, very sensitively using transverse susceptibility. And this is something that I will introduce because my goal is to not just talk about magnetic particles for biomedical applications, but how to control or engineer the anisotropy for that purpose. Okay. The second experiment that I'm going to focus on today is this one, SAR for hyperthermia. SAR is specific absorption rate. It's an acronym for specific absorption rate, and that is a measure of the heating efficiency. In fact, I'm going to tell you that we can take magnetic nanoparticles and effectively heat up the cancer cells and kill them by that process. Okay, so those are the 2 experiments. We are also active in other areas. I'm not going to go into that. Again, a quick shot of basically what the lab is all about. With some pictures here. We work on nanoparticles of various kinds. For example, this is one of our workhorse. This is a Dynacool physical property measurement system. It's a very versatile magnetometer from Quantum Design, and we use this. And you can see there's a vibrating sample magnetometer VSM on top that is always working. Looking at measuring magnetic properties, and we work on a number of typical areas, and these are some examples. And I'll get to that later on when I discuss specific
just something
types of particles.
Just one thing.
This is the outline of my talk after a rather lengthy introduction into the areas. So basically, what I'm going to talk about is it's all in Portuguese, so I have no idea what I'm going to talk about. Hopefully, I will make some sense because the first thing I'm going to talk about is an introduction to magnetic anisotropy, some of the basic and fundamental ideas behind what it is. What is anisotropy? The main part of the talk is going to be applying anisotropic nanoparticles for biomedical applications. I don't know if you're familiar with an American TV show. It's a reasonably popular here. It used to be. It's called Dancing with the Stars. So today I'm going to do a play on words. I'm going to talk about dancing with the SARS. Remember, SAR is specific absorption rate. Unfortunately, SARS is not a good acronym now, since now we're dealing with coronavirus. You know, SARS used to be another, bad virus a few years back. So it's not that SARS. The SARS that I'm referring to here specifically is the heating efficiency. It's the figure of merit called specific absorption rate. Okay, so my whole idea is to tell you that how we can go beyond the state of the art. We can go beyond what is commercially available out there. We can do it we have done this in our research lab by playing around with the anisotropy in nanoparticles and improving the way in which you can heat up and use it for cancer therapy. So that's the big picture. Okay, so what is magnetic anisotropy? Of course, if you want to talk about magnetism, then you have to go all the way to quantum mechanics, and we don't have time for that today, right? This is just a 1-hour talk. So basically, I'm going to start somewhere to give you an idea of what anisotropy is all about. So let's start with the magnetization vector. I'm sure some of you students who took quantum mechanics or some basic solid state have learned about some magnetism in your classes. So essentially, a magnetic material can be magnetized, and what you see here is the magnetization vector, M. You apply a magnetic field, and the interaction can be represented by a very simple form here for the total energy. It has 2 terms in it. It could have other terms, but this is the very simplified version. And the first one here, you can see that it depends on the external field, and so this is called the Zeeman term. So essentially, the strength of this energy is determined by the applied field. The second term here is what is relevant to us, and this term is the anisotropy energy. So in this case, it's a very simple term, K1. It's a uniaxial term because, guess what? Nanoparticles typically are spherical, and you really don't have a preferred direction. So that's what we use in terms of a simple way to represent the anisotropy energy. So that's uniaxial anisotropy. So as you can see, it's a competition between the Zeeman term and the internal energy, or the anisotropy energy, in any given magnetic materials. So when you apply the magnetic field, of course, there's going to be a competition between these 2 energies. You can minimize the energy. I know you can do calculus, right? You can minimize the energy. And essentially, this here determines the shape of the magnetization curve. And this energy difference is what determines whether you can magnetize a material very easily at small fields, or it takes a much larger field. And this is nothing but the easy direction and the hard direction, right? So that the shape of the MH curve, or what we call the hysteresis loop in a ferromagnet, depends on these competing energy scales, and it depends on anisotropy. Now you can see why I'm leading up to the fact that anisotropy is the most important quantity that you can tune in a ferromagnetic material to be able to change the magnetization behavior. And that is important for this application today. Okay, so this is uniaxial. We can also take into account higher order symmetries, cubic, for example. By the way, I'm a physicist, so I have to show at least one equation. But believe me, this is probably the one, or maybe I have one more slide with an equation. That's it. No other equations in this talk. All right. So we can take cubic or any other kind of anisotropies. We can deal with that. However, when it comes to nanoparticles, you have to think about several aspects that can contribute to this effective anisotropy. Let's start from the left here, the surface. I don't know how many of you are working in nanomaterials, but if you are, guess what? You are all surface scientists. The reason I say that is because that in nanomaterials, it need not be just magnetic. It could be any kind of a nanoparticle. The surface atoms are the most important because they determine the configuration as well as the physical properties of the materials. So they are the most important, okay? So by manipulating the surface atoms in nanoparticles, we can effectively change the magnetic properties, certainly the anisotropy. So that's one thing that can contribute to this. Now shape, the second one, shape is easy to understand. For example, you can make nanotubes or nanowires, and obviously there is a preferred direction here. You have the longitudinal direction of the wire or the tube, which is the EC direction. That is the EC direction in which you can magnetize the the material. So shape, it's easy to understand that you can make different shapes and you can change the effective magnetic properties or the approach to saturation, as you can see in the curve. Strain is important. You can actually use, for example, in thin films, you can, the magnetization, if it's a magnetic film, the magnetization, the magnetic properties are determined by the interfacial strain. This is the elastic contribution, magnetoelastic contribution, and you can use the mismatch in the lattice and the strain to be able to control the magnetic properties. So strain can play a role too. Configuration is something interesting. I'll come to that towards the end. You can actually combine different kinds of nanoparticles to make clusters, and you can make very specific geometric clusters and use that for manipulating the magnetic anisotropy and magnetic properties. And last but not the least is unidirectional or exchange anisotropy. Now, exchange coupling is extremely important for the spintronics community. I know that there are groups at Unicamp in your institute working on spintronics area, all right? Now, it turns out that exchange bias is because when you take a ferromagnetic material and a combination of an antiferromagnet and a ferromagnet, now at the interface, depending on the difference in the anisotropy of these materials, you can actually have the spins at the interface pinned by the bottom layer, okay? And this is the so-called exchange bias. And essentially what it does is that you can think of the loop itself being shifted. This is called the exchange bias, depending on this amount of energy at the interface. And the coercivity can increase or decrease for a hysteresis loop. And that is because of the so-called exchange bias. Now, exchange bias is extremely important for spintronic devices like MRAMs, spin valves, all kinds of spintronic devices. This is a very important phenomenon, exchange bias. Today, I'm going to tell you that exchange bias is equally important for nanomedicine, for cancer therapy applications, okay? All right. So one of the things that we can do is, for example, consider why should we look at anisotropic nanoparticles? Why can't we just make nice, perfect spherical nanoparticles? Well, guess what? It turns out that making perfect symmetric particles is actually more difficult. You can actually make easily anisotropic nanomaterials, okay? And not only that, anisotropic nanoparticles are there everywhere. It's omnipresent, okay? To show this, I'm going to show you a series of TEM images. These are transmission electron microscope images that many of you, obviously, if you're working in nanoparticles, you take it to a microscope, electron microscope, and look at it with a TEM. So what you're seeing here are three images. So 1, 2, 3, and they are the same material. They are Fe3O4, magnetite or ferrites, okay? Spinel ferrites, magnetite. It's the same material, but where the source of these nanoparticles of these images is what makes them interesting, okay? Let's look at the one here on the left, the first one here. It turns out that what you're seeing here, these are magnetite or, Fe3O4 magnetite nanocrystals that are lined up into nice little chains here using dipolar coupling. And it turns out that this is formed naturally in a biological system in bacteria. These are called magnetotactic bacteria. Turns out these magnetotactic bacteria produces these magnetite nanocrystals, chains them up. And do you know why they're chaining them up? They're doing it to make tiny compasses, just like the Chinese did when they first discovered the first magnetic material known to man, which is called lodestone, which is nothing but magnetite, Fe3O4. So what these bacteria are doing what the Chinese did, the Chinese explorers did. They're making tiny navigational compasses, and they use it for orienting themselves to go and search for food, okay? So they're making tiny compasses here, using nanoparticles. The second example, the one in the middle here, is out of this world, as we use this expression here in the US commonly. And literally out of this world, because what you're seeing here, these are meteorites, these are rocks from Mars. These are meteorite chunks. These are iron oxide nanoparticles magnetite nanoparticles that are found in meteorite chunks that crashed into the Arizona desert, and they are supposed to be from Mars. So you got nanoparticles in life forms, nanoparticles in rocks from outer space, from different planets. Now, does that mean that is that life on Mars? No, I'm not going to talk about that. That's for someone else to look at. But just to give an example here, look at the facets, the surface facets. These are faceted nanoparticles. You can see that they're highly anisotropic. And again, this whole configuration, this chain is highly anisotropic. Okay, so I'm giving you examples of how anisotropy is existent in nature and how it is there in biological systems and in rocks. By the way, here's a little piece of history for those of you that are interested in nanomagnetism. If you want to look at when it all started in the 1930s, interest in this field. And the first scientists that actually started looking at nanoparticles, magnetic nanoparticles, they're not physicists, they were not chemists or engineers, they were geologists. So most of the early studies, in fact, the word superparamagnetism, all these things were coined by geologists. So next time when you go out, in your campus at Unicamp, you meet a geology friend in geology, give them, your best wishes and thank them for introducing the field of this exciting field of magnetic nanoparticles and nanomagnetism. Okay, so third example is actually man-made. These are, again, magnetite or ferrite nanocrystals. They were produced using a microwave plasma technique, and they were done at Carnegie Mellon University in Pittsburgh by a good friend of mine, Professor Mike McHenry and his group. And here you can see between these two, you can actually see that they look pretty similar. The ones from the meteorite and also the faceted nanoparticles, which are also highly anisotropic. They can actually be produced in the lab using various techniques. Okay, so again, the idea here is to show you that anisotropy is there everywhere. So let's try to put it to good use. Okay, let's not strive for symmetry. Let's strive for anisotropy, because that's more important for applications, for changing the magnetic properties, and for many different applications. All right, so I'm also going to tell you how we can measure anisotropy, because anisotropy is so important. Here, I would like to introduce a technique that we developed in my lab many years back, but it turns out to be one of the best ways and the most sensitive way of measuring the magnetic anisotropy in nanoparticles, as well as in films or any kind of materials, any kind of magnets. What we do here is that we use a transverse susceptibility, and the transverse is based on what is known as tunnel diode oscillator, TDO. Okay, so essentially what we do is we build oscillators. The sample is sitting inside an inductive coil here, and it's an LC circuit. It's nothing but a Tench circuit. Students would have learned about AC circuits, LC circuits, resonance circuits. Okay, so this is an LC circuit. Now it is driven by a tunnel diode. Now, if you remember your tunnel diode characteristics back from AC circuits, you know that there is a negative resistance region, right? In fact, for a tunnel diode, if you increase the voltage, the current goes down and then goes up. Now, if you bias the tunnel diode in the negative resistance region, it acts as a power source for this LC circuit and sustains the oscillations. Basically, it's an ultra-stable self-resonant circuit that we build to measure the susceptibility. The sample sitting inside here, and of course, this whole thing goes into a superconducting magnet, the PPMS that I discussed earlier. So that's, for example, how the PPMS sample probe looks like. So this is our coil right here. And all we do, and of course, the transverse geometry is because the DC field supplied by the magnetometer, the PPMS, is perpendicular to the small modulating RF field here on the sample, and that sets up this crossed field geometry, and from which we can extract the transverse susceptibility. Now, it turns out that the transverse susceptibility, since it's a resonance circuit, all we do is monitor the shift in the resonance frequency, and from that, we can actually get a change in the transverse susceptibility. Now, I'll tell you why transverse in the next slide, but here's an idea, here's why this is a very good technique, because the sensitivity is very high. For example, all we do is measure the shift in the resonance frequency. You can use a frequency counter to measure the frequency. By the way, those who are experimentalists should know that frequency is perhaps the most accurately measurable quantity. Ideally, you want to measure any kind of physical property as a frequency measurement experiment, okay? So you gain from the sensitivity in measuring frequency. In this case, we can measure the frequency shifts of the order of 10 hertz in the overall resonance of 25 megahertz. So you can do the math. You can see that you can get parts per million resolution when you want to study very fine phase transitions, whether they are magnetic field induced or temperature induced. So this particular technique has been extremely useful in studying anisotropy fields and other kinds of phenomena, switching and everything with a high degree of sensitivity. Okay, we can also do this over a large temperature range using our low temperature superconducting magnet. We can apply fields. So right now we can apply up to 10 Tesla in our lab, minus 10 to 10 Tesla. So we can actually look at a whole range of parameter space. Now the question is, why do it transverse? Why not do conventional standard susceptibility like you would do in a squid magnetometer or in a VSM, for example? The reason why we set this up is due to an old paper by a very famous theorist back in 1957, Amika Maharoni. He's from Israel, from Weizmann Institute in Israel. He published a paper where what he did was he took a single domain nanoparticle. It's a theoretical calculation, T equal to 0, ideal calculation. And he plotted, this is from his original paper from 1957. Unfortunately, it was published in the Bulletin of Israeli Physical Society. It's hard to get, but we were able to get that. So this is the actual result here. So what he plotted here is that he analytically calculated both the parallel component and the transverse component of susceptibility. You can see that the parallel component is a conventional one, right? dm by dh in the same direction. But when you apply fields in the crossed field direction, transverse direction, this is actually the transverse susceptibility, and you see that it's a tensor, okay? That's what we are trying to measure. In fact, what he pointed out is that here's a plot, reduced field with respect to the anisotropy field. If you go from positive saturation to negative, for the parallel susceptibility, as you would expect, you see a singularity. This is like taking the first derivative of your hysteresis loop, okay? So it means when the spins are switching, when you change the sign, change the polarity, you get where the maximum slope changes, you get a peak. So it's a singular point detection. Now he pointed out that in the transverse configuration, in addition to the switching field here, you also see two other peaks which are symmetrically located on either side, and it turns out that they're located at the anisotropy field. Remember that anisotropy energy is one of the most important fundamental energy scales in ferromagnets. So this is a way of identifying the anisotropy field directly by looking at these sharp peaks. That's exactly what we did. We tried to set up an experiment to measure what Maharoni predicted all those years back, and then we were successful in doing that. And this is one of our signature experiments that made us, really well known in the community, in the magnetic nanoparticle community, because we can measure these anisotropy peaks very, very precisely. Yeah, we've been looking at this for many years on many kinds of magnetic particles, magnetic films. So you can see that this is our data here. This is for a manganite film. Because it's a thin film, the axes are well defined, right? The anisotropy is well defined. So you can see the peaks are very sharp. And you can actually find out what the anisotropy field is, and you can, from that, you're putting in the geometric factor, you can calculate the anisotropy constant from this thing, and everything goes well. Okay, so we've done a lot of work on many different particles. These are Magnemite nanoparticles. It's not on there, but I can tell you these are gamma Fe2O3 Magnemite nanoparticles, roughly about 15 nanometers. And you can see that because we do this experiment, the transverse susceptibility in a PPMS, we can not only change the field, we can change the temperature. So we can actually track this whole thing as you go from the super paramagnetic regime at low temperatures to the blocked regime. Okay, so we can completely analyze the magnetization dynamics RF at high frequency, basically over the entire HT plane. And that's the power of this experiment. Okay, so by now, hopefully I've convinced you that anisotropic magnetic nanoparticles are very important. They're there everywhere. And we also have ways to measure anisotropy very precisely. Okay, you can also do this by other experiments, ferromagnetic resonance, FMR,
BRUA light scattering, BLS. There are many ways of measuring anisotropy, but today, hopefully I told you something that we do, which is unique to my group, where we use transverse susceptibility to measure anisotropy. So now I'm going to switch gears and go towards the bio area. So what can we do with these magnetic nanoparticles, these anisotropic nanoparticles? Can we apply them to biomedical applications? And here are some ideas, some areas where magnetic nanoparticles are already being applied. Okay, so I'm going to start with, let's start with this one right here, targeted drug delivery. Now this cartoon shows that basically what you can do is that you can actually take magnetic nanoparticles and use them as navigational chips. You can actually functionalize them. That's the nice thing about nanoparticles, because they have a lot of nice surface area, large surface area, high surface energy. You can actually functionalize them using, you can actually make them biocompatible. You can also use ligand chemistry to attach various molecules. And so these magnetic nanoparticles, because they are super paramagnetic, by applying an external magnetic field, they respond to it, and you can actually drag them inside the human body. Okay, so these magnetic particles can be used as navigational aids, as like ships carrying the drugs, and you can deliver them locally to the infected area. All right. There's also some work we're trying to initialize right now of using magnetic particles for combining, attaching various antibodies to magnetic particles, and then looking at the reaction with the COVID-19, virus, especially the spike proteins, but I'm not going to go into that. Okay, that's something new that we're thinking of right now. But the whole idea here is that the particles can be moved within a magnetic field, and because they are super paramagnetic, if you remove the field, the particles become demagnetized. Okay, so that's very advantageous. We don't work in this area of targeted drug delivery. However, there are many groups that are looking at this. Now, another area where magnetic nanoparticles are used quite effectively, and my group has also made some contributions here, you can actually find out that magnetic particles can improve the MRI contrast, image contrast. MRI is magnetic resonance imaging. It's one of the most common techniques that you see used in hospitals. Basically, it's based on NMR, nuclear magnetic resonance. The moments are very small because these are nuclear moments, much smaller than the electronic moments, right? So you need high fields, large magnetic fields for the conventional MRI. Now, it turns out that the way it works is that the water protons here, they're set into precession by applying a AC and DC field. And then when you remove the field, the precession, basically, it starts deprecessing. The precession goes dephasing. And from that, from the time of dephasing, you can actually get these kind of images, which are the well-known MRI images. Now, if you put magnetic nanoparticles inside the area that you want to image, like the cell that you want to image, now it turns out that the fringing fields from the nanoparticles actually interfere with the T1 and T2 relaxation times, NMR relaxation times. And because of that, because of the difference with and without the nanoparticles, you can actually get a better contrast. So it has been demonstrated. This is an area there's been a lot of work, but it's clearly shown that magnetic nanoparticles are very useful for improving the contrast in MRI images. More recently, there's a very exciting new area here. This is magnetic particle imaging, MPI. So what is the difference between MRI and MPI? Remember that MRI works on nuclear resonance, right? It works on basically nuclear spins interacting with the local, tissue. Now, in this case here, this is actually imaging the particles themselves. So the signal is much better, much bigger, much brighter. So MPI is actually right now, there's a company in Berkeley, based in California, that is actually selling MPI systems. These are very similar to the MRI systems, but they have much better contrast. And this is again a case where magnetic particles are really making an impact. There's also cell separation. I'm not going to go into that because of lack of time. But I'm going to talk, for the rest of the talk, I'm going to tell you how we can use magnetic nanoparticles without attaching any other drug molecules, just particles themselves. Can we use them for therapy, cancer therapy? Yes, we can. And this is what we do. We can locally heat the nanoparticles, and this is called hyperthermia. The rest of my talk, I'm going to tell you how we can use these magnetic nanoparticles for hypothermia. We can also think of various things. We can combine magnetic particles with other kinds of nanoparticles. So this here is a broad, diagram that shows various kinds of chemistry and structures, biological interfaces that can be used, and different kinds of coatings and functionalization. There's a lot of surface chemistry that's involved if you want to apply nanomaterials for biological applications. You have to, again, look at the size of these particles, and that depends on the compatibility and also the kind of biomedical application you have in mind. And finally, most importantly, you can also think about making particles of various shapes. These are typical shapes that you can think of, and they are also important for specific applications. Okay, so cancer, well, I don't need to basically say why it is important to find treatment for cancer. Well, right now, apparently cancer has taken a backseat because of COVID-19. Everybody's now focusing on that, but cancer has always been around as one of the diseases that need to be controlled. Here's a world map. Of course, your country is right there. Okay, so you do have a cancer problem too. But essentially, we need to look at all kinds of basically ways to treat cancer. Now, many of you are maybe physicists or material scientists, and you may be wondering, oh, cancer treatment, that's the job of the doctors, the medical doctors. Now, that is not the answer right now. It turns out that all of us can combine our expertise, physicists, chemists, and we can all come together to address this very important challenge of finding new ways of treating cancer. Okay? Just to give an idea, this is the Physics Today, July 2019. There's an article here which tells you how mathematical modeling has played a role in understanding the process of metastasis. Metastasis is the process by which cancer cells grow, they get bigger, and they leave the cancer region and go to other parts of the body. It's very important to understand the mechanism. So this one tells you that physicists and physicists and mathematicians have been very, played very important roles in understanding this process of cancer development or cancer growth. Okay, so now let us come back to using magnetic nanoparticles and how do we treat cancer, how do we kill cancer cells? So we do this by this technique of hypothermia magnetica, magnetic hypothermia. And I am not a biochemist, so this is what happens. It turns out that, I'm sorry, hold on, let me go to the laser pointer, yeah. Okay, so basically what we do is that we take these magnetic nanoparticles and we subject them, instead of a DC field, we subject them to a high frequency AC magnetic field, and that generates heat. I'll explain to you how this is done, okay? I am not a biochemist or a biophysicist, but this is what happens. It turns out that when you apply an AC field, you have protein denaturation. And essentially, the idea is to break the cell membrane of the cancer cell. That's the goal. The way to kill cancer cell is to break the cell membrane, to destroy it, some way or the other, okay? So cell membrane deactivation, and eventually that causes the cell to die. We call this process as apoptosis. Now, apoptosis is, if you may think of a friendly, benign way of killing cancer cells. We do this by generating local heat. Yeah. Friendly way of killing cancer cells is by locally heating them up. And you don't need to heat them up a lot. It turns out that the window is 40 to 45 degrees Celsius. It's only just a few degrees above the human body temperature. You need to heat up the cancer cells to this temperature range, 40 to 45 degrees Celsius, and you need to hold it for some time for the cells to get hot and they die. Cancer cells don't like heat. They don't like Florida in summer, for sure. Okay, so we are going to generate local heat by using magnetic particles by applying a high frequency AC magnetic field at large amplitude, field. Obviously, you need a large amplitude. You need to saturate the particles and do it at high frequency. So that's the technique here. Now, there's a company in Berlin, in Germany, that has actually come up with this nanotherm therapy. So they basically inject the nanoparticles, take it to the tumor area or inject directly into the tumor, and then they apply an AC field So it's like you can see it's like an MRI system. The patient is going through that, through the magnetic field, through the AC magnetic field, and then they have actually been successful in showing that it is very effective in killing cancer cells. Okay, let's get back to some physics. How does this work? The reason why I spent a long time talking about the hysteresis loops and the shape of the hysteresis loops is because that is the key here, hysteresis. So hysteresis is basically dissipation. The area inside a hysteresis loop is nothing but dissipation, and dissipation is our loss of energy, and the dissipation here is in the form of heat. So we need to optimize the hysteretic losses. Now, if you look at this one right here, don't think of the magnetization curve for nanoparticles that you can measure in a squid or a VSM. That is the DC curve, right? So that one, obviously, if it's super paramagnetic, there is no hysteresis. Here, what we're talking about is the AC response. So this hysteresis here is to the high frequency of the field. I mean, think of it this way. Each of these magnetic particles, they are a large super paramagnet. They're like almost like a single ferromagnet, small ferromagnet by themselves. And when you apply a high frequency AC field, the magnetic moment tries to follow the high frequency, but they cannot, they go out of phase. And because of that, you can generate these kind of hysteresis loops. And this is what we want to optimize or maximize, because the specific absorption rate, like any other experiment, you need a figure of merit to find out what it is that you want to improve. And the figure of merit is the specific absorption rate, which is nothing but the area multiplied by the frequency. And you need to optimize this, keeping in mind that you need to get the highest SAR with the smallest number of particles, because remember that you're going to inject these particles inside the human body. So obviously it's not going to you have to be very careful about how much you put in. So the next few minutes, I'm going to talk about how can we optimize these parameters with the nanoparticles. I'm going to skip the slide. Again, this is for physicists. If you're interested, there are different time scales involved. And you can see this is the size dependence, magnetization. So one of the ways to improve the heating efficiency is to produce magnetic nanoparticles with large magnetization. However, it's very difficult, as you know, when you make particles smaller, when you make a ferromagnet, make it into a nanoparticle, the magnetization goes down. And this is because of the surface being, disordered, having disordered spins, okay? There is also a critical size dependence for improving the SAR. It is also called SLP. SLP is specific loss power. It's the same quantity. It's the heating efficiency. You can see there's a size dependence, there's an optimum size. Now, it turns out that the best size for these kind of cancer therapy is somewhere between 20 to 35 nanometers. Not smaller and not bigger, if you look at this curve. For health reasons, you cannot use very small particles for hypothermia treatment, okay? And this is because if you take nanoparticles less than 10 nanometers or 5 nanometers or less, the chances of these particles penetrating the so-called blood-brain barrier is very high. So which means these particles end up going to the brain, the wrong place where you want these magnetic particles to go. When you inject them into the human body, you want them to do their job, kill the cancer cells, and then basically go down south, flush out of your system, go down south to Florida, okay? So that's what you want. So you don't want to, for health reasons, it's very risky to inject very small particles. So there is a limit. You cannot, use particles that are really small for hypothermia. So we have to be careful about that. Okay, so how do we measure the magnetic hypothermia in the lab? So we do it in two different labs. One of them is in our lab, and we do this here. This is our setup right now. I'm sitting in my office, so it's about 100 feet from me on the other side of the room, on the other side of the building. Okay, so we have it set up, and what we do is a calorimetric method. So we have our nanoparticles, and I'm going to show you some nanoparticles in the next few slides, the kind of anisotropic nanoparticles that we make. So we put these particles inside the inductive coil here. That's the AC power supply. It's a commercial one. Remember that we need to have large amplitude, high frequency power supplies. So it's not normally most of the AC power supplies, the amplitude they produce is very small. So we need much higher amplitudes here. Okay, I'll give you the numbers in a minute. And so what we do is that we measure, we have these particles in fluid or any kind of medium here, and we directly measure the temperature using various kinds of thermal sensors. It could be optical, using a laser, we can measure the local temperature. So what we measure here is the rate at which the temperature changes. Keep in mind that we have to heat up the cancer cells, right, to above 40 degrees. So we measure the effectiveness by how fast we can increase the temperature of the nanoparticles. Now we also take our samples and ship it across the Atlantic to Spain, to a beautiful Basque country in northern Spain, in Bilbao, where at the University of Basque Country, we have a close collaboration with several group members there. And what they do is that for the same particles, they measure the AC magnetometry. They directly measure the hysteresis loop. But in our case, the data looks like this, okay? So this is the temperature, function of time, and we are just measuring at different AC field amplitudes. In our case, the frequency is fixed. It's 325 kilohertz. It's a fixed frequency power supply. And again, the numbers are important. I'll tell you why. And here are different AC field amplitudes. Now from this, you can see that that's the hypothermia window, right? 40 to 45. So ideally, you want the heating to be really fast and then basically stabilize in this window. You don't want to overheat it. There's no reason for that. And if you don't apply enough field, you will never get here, okay? But by looking at, in this case, these are manganese ferrite nanoparticles. For different kinds of particles, you can find out exactly what are the parameters needed, what frequency, what field is needed for it to stabilize or saturate in this temperature. By the way, this saturation is simply because of, heat exchange, latent heat, and the specific heat, of the medium in which the cells, in which these particles are. Now, what our colleagues do in Spain is they actually measure the AC hysteresis loops, and they can actually measure the hysteresis curve here inside. They can get the SAR from that, okay? Two different ways of measuring SAR. Now, I usually tell students a little story here, basically saying that if you want to publish a paper in a high impact journal, physical review letters or Nature Materials or whatever it is, ideally you want to prove whatever result that you want to show. You want to prove this by doing two different experiments converging to the same result, okay? Now that's what we're trying to do here. The same quantity, SAR, by two different experiments, and not only two different experiments, two different labs, two different countries. So the message here is that we need to really establish international collaborations where we bring together different expertise and experiences, and hopefully someday we will be able to do this between USF and Unicamp. I look forward to that. Okay, now I say that there's several hundred kilohertz and a few hundred Oersted here. This is the number you have to see here, 200, 400, only that. Why is it? Why can't we just increase the frequency to hundreds of kilohertz or even go to gigahertz? We can do that. Why can't we do it? And why can't we increase the applied amplitude, field amplitude to Tesla fields? The answer is, remember, this is treatment for humans. So we have to work with the condition of the human body and the limits that the human body can withstand. You cannot arbitrarily apply very large fields and very high frequency, because you will cause nervous stimulation. You'll have paralytic attacks, okay? The nervous system will be in trouble. So one has to be very careful, and this is why for the magnetic hyperthermia community, you have to work with these fields, these limits. The limit here is the product of the field and the frequency, and in these units, in SI units, it's that. Think of it as some kind of a pain threshold, okay? So basically, think of it as if you go to a hospital, right? If you go to the emergency room, this is what the person that first meets the patient here in the US, and I'm sure it's the same in Brazil, is what we call a triage nurse. And the triage nurse, she will ask when the signs the patient in, she will ask a question on a scale of 1 to 10, what is your pain level? And of course, the patient will say 2 or 5, or if they're close to dying, they'll say 8, right, or 10. So that's what we're talking about here. So basically, the conditions, remember this treatment of applying high field and high frequency to the nanoparticles to the human inside the human body over a certain period of time for the treatment to be effective. So one has to keep in mind the condition of the patient, whether this patient can withstand these conditions, and so the field and frequency limits are set below certain values. Okay. All right. So now I'm going to talk about what can we do here. I know that we started a little bit late, and I'm running a little bit late. So may I ask Fanny if she's has her microphone on? Will it be okay if I take about 10 or 15 minutes? Or
yes, I guess there's no problem. You can continue.
So that'll help me able to complete some of the ideas that I wanted to present today. Okay, so thank you. So let's get back to now. How do we optimize the SAR, right? Remember, that's what we need to optimize, the heating efficiency. So we can do that in many ways, or think of many ways. You know that it's dependent on the saturation magnetization. Clearly, the magnetization of the nanoparticles, the higher it is, the more efficient, the higher the SAR is. Now here's something that shows the SAR in watts per gram, okay, because it's a dissipation, it's a power. And you can see for iron oxide, magimite in this case, is very low compared to iron cobalt. Iron cobalt is even better because the magnetization is very high. Unfortunately, iron cobalt, they are not allowed inside the human body. Okay, only iron oxide, whether it's Fe2O3 or Fe3O4 magnetite, these are the only magnetic materials you can actually, they're considered biocompatible. They're approved by the FDA, the Food and Drug Administration. They've been authorized for these kind of therapies. Okay, unfortunately, even though iron and iron cobalt is much higher saturation magnetization, we still have to mostly work with this, somehow improve this. Okay, that's the only thing that's allowed. I already talked about a size dependence, but can we do something else? Can we change the shape of these particles? Can we change the surface structure of these particles and magnetic oxide or other kinds of particles? Can we make some kind of core shell structures, some kind of hybrid geometry where you have a core and a shell of different materials, and you can have exchange interaction or exchange anisotropy between the core and the shell? These are the things that we were interested in my lab. And a few years back when we started this work, we found that not many groups were looking at, magnetic hypothermia groups were looking at anisotropy. They were just getting commercial samples, commercial particles, and doing the experiments. We thought that we could improve this by making a careful study of anisotropy and the role it plays on improving the SAR. Okay, but first, when we saw this one right here, we thought, okay, we thought of an idea. We thought, okay, how about fooling the cells? How about fooling the cells by having the core, making a core shell nanoparticle with the core being iron, which has a large saturation magnetization, and the thin shell completely covering the iron is an oxide magnetite, which is known to be biocompatible. So in other words, we try to make these core shell, iron oxide shell, iron core nanoparticles. And here is something from our lab. You can see they're beautiful core shell nanoparticles. The darker one inside is the iron. The lighter shell is the iron oxide. And we thought we can fool the cells because clearly iron oxide is biocompatible. So the cell will think that it is biocompatible, no problem. But inside we have the iron, which has a larger saturation magnetization. So we did this experiment, and initially we were successful. We were very happy. However, our happiness was short-lived. Okay, I'll tell you the story before moving on to the anisotropy stories. Okay, so for those of you that are chemists or who do chemical synthesis, so this is basically what we do typically. I want to keep this very simple. It's chemistry in a beaker. Essentially, this is the kind of setup that we have, 3-neck flask and everything. We do reactions in there, and the particle size is determined by the temperature, the time, and the surfactants that you use, so which control the growth, okay? And also the various recipes. So it is more like an art, not even a science. Some of my students and postdocs are very good at it, and they have somehow developed this instinct of making very high quality nanoparticles. Even though we are a physics lab, we have a lot of chemistry going on, chemical synthesis going on in our labs. Okay, so there is a problem. And this problem, when you make these core shell particles, this problem is known as Kendall Effect. So that back in the 1950s, chemical engineers and chemists knew this problem. And basically, the problem is very simple. Metallurgists actually knew this problem. By the way, the term metallurgy has disappeared. When I was a student, PhD student, and I'm not going to tell you that it was long ago, there were departments of metallurgy in many countries. You know, I actually did my PhD in India, so we had departments of metallurgy there in some of the schools. But now it's all become material science. You don't find any department of metallurgy anywhere in any country. Okay, so it turns out that if you think of the metal core and the iron core, iron oxide shell, there's a problem because it turns out that because of the differential diffusion of the metal ion versus the oxygen ion, because of this, there are instabilities at the interface, which causes these holes, the voids, to open up. These holes get bigger and bigger, and finally, what happens is that the iron inside oxidizes, leaving completely whole inside, and the iron oxide shell gets thicker, so you get beautiful hollow nanoparticles. So if your first intention was to show lovely hollow nanoparticles, this is the way to go. Make core shell particles and let Kendall effect play the role to convert it into hollow nanoparticles. Unfortunately, this is not good for our application because we want these particles to be stable over time, because we want them to be effective for hypothermia, to have a large star. So we did the study and we looked at all the intermediate, kind of particles. We looked at the magnetic properties, and we also looked at, SAR. Let me show you some results. So here are some SAR results from our lab. I already mentioned to you how we do 2 different experiments to get the SAR. So let's look at this. Basically, we were very happy because if you compare the core shell particles with the iron core and the iron oxide shell of different sizes compared to the fully solid iron oxide nanoparticles, the SAR is much higher. Okay, I'm going to show the SAR in histograms for easy comparison. However, our happiness was short-lived because if you look at this as a function of time, we did this over many days and weeks. It turns out that because of this Kendall effect, the core shell particle was continuously transforming into finally giving you a hollow particle. And of course, when it becomes a hollow particle, it doesn't heat up anymore. The SAR is non-existent. Okay. Unfortunately, even though the proof of principle worked, we were able to increase the SAR beyond the conventional solid magnetite particles that are always used. Unfortunately, it's not effective. It's not going to be very effective. So we thought that this is done, but we liked the idea of core shell. So we thought, what can we do next? And this is when we actually found this paper. This was back in 2011. There's a group from Korea, it's a very good group in Korea, that published a paper in Nature Nanotechnology showing that if you have core shell nanoparticles of 2 different ferrites, in this case, they use cobalt ferrites, manganese ferrites, there is no Kendall effect because it's not metal oxide, both of them are oxides. So it's all oxide core shell particles. And they actually showed that the SAR is very high. Okay. So I thought, okay, well, that goes my idea. We tried to see if we can do this, but then I looked at the materials. Saw the first element, cobalt. Cobalt is poison. It's impossible for FDA to give you an approval for cobalt-based materials. Okay, so the best chance of doing anything to get through the system, to get FDA approval and to go for clinical trials is to make sure that we work with biocompatible oxides that are already approved by the FDA. So we have to make a core shell particle based on Fe3O4, but all oxide, okay? And so I gave this as a challenge to one of my postdocs, and she came back with this. She did it. She made exchange-coupled core shell iron oxide, 2 different phases of iron oxide. FeO is a Wüstite phase. It's actually a lesser known phase of iron oxide, and it's antiferromagnetic. Now that's our core. And remember, our outside has to be biocompatible, right? So it has to be magnetite. So magnetite is the shell. So it's sort of an inverse core shell approach. The core is actually antiferromagnetic and the shell is ordered, okay? Usually it's the other way around. When you take nanoparticles, magnetic nanoparticles, the core is ordered and the shell is disordered, you know, because of the spin disorder at the surface. Here, it's the other way around. It's an inverse core shell. And the other thing is that because it's an antiferromagnet, this is a tiny spin valve. It shows exchange bias. There's the data. We published this stuff, 5 years back. You can see the loop shift when you do the exchange bias experiment by cooling the particles down from above the Néel temperature down, and looking at these curves. So these are tiny nano spin valves showing exchange bias. And guess what? This is just to show you how we can play with, yeah, okay. Yeah, here's another story. This postdoc, her name is Hafsa Khurshid, and after she did these experiments, we changed her first name from Hafsa to Hermione. The wizard in Harry Potter, the lady, the girl. Okay, so basically what she did was amazing. She came to me and said, okay, Harry, I thought you wanted to change the anisotropy. That's our goal, right? And I said, yeah. So she said that I'm going to try to make core shell particles, FeO, Fe3O4, all oxide, no Kendall Effect problem. At the same time, I'm going to change the shapes of these core shell particles. And I was like, okay, I'm not a chemist. So I said, good luck with that. And she went and came back with beautiful shape anisotropy. In other words, she came back with, I call them nano Legos. Somehow she was able to play with the chemistry, and the same system, she made spherical particles, she made cubes here, and she made these star-shaped particles. So in this case, we can actually compare the surface anisotropy, how it affects the magnetic properties as well as the SAR. This is one of the most important slides of this presentation. We were one of the first groups that actually showed very clearly how shape dependence and surface anisotropy can completely change the SAR, can improve the SAR. It's very compelling. Spheres, cubes, octopods. In all these cases, we made sure that the geometry, the physical geometry, is rescaled, because obviously the volume of a cube and a sphere, they're all different, right? So we have to make the shapes and the sizes accordingly. So we made sure we took care of that, scaled them. And look at this data right here, histogram. The comparison of the SAR values for spheres, cubes, octopods. There is no question here. It's very clear that as you increase the effective anisotropy, in this case the surfaces, you get a higher SAR. It's one of the most important results that came out from our group during a few years back, when people are not really looking at it, not paying attention to this. And we were the first group that pointed out the importance of anisotropy. And now I think many groups are now trying to make sure that they look at the shape anisotropy and the effects of this on the SAR. So this is one of the first results that came out. We got pretty excited, and I said, okay, let's go back to those chains. Remember those bacteria, the magnetotactic bacteria, which automatically, naturally forms these dipolar coupled chains? So we wanted to look at this, and in collaboration with the Spanish group in Bilbao, this came out last year in small. So we actually found out that indeed we can. Oops, I'm sorry, going the other way around. We can actually increase the SAR. Okay, so this was done in collaboration with them. And then there's another postdoc in my group, Raja Das, who decided to mimic these chains, these short chains in the bacteria. And instead of having chains which are weakly coupled, right, dipolar coupling is not very strong, it's weakly coupled. Instead, he wanted to make nanorods. These are robust nanorods of magnetite. I would say some of the best nanorods that you will find out there. And the best way to make them is using a hydrothermal technique. This is like a pressure cooker. It's a pressure cell. So you basically cook these nanoparticles at high pressure. In addition to temperature, time, surfactants which control the growth of these nanoparticles, you add another variable, which is pressure. When you do that, you can actually control the aspect ratio. You can produce really good nanorods. I'm going to go fast because I need to end my presentation and take some questions if you have. So we looked at several aspect ratios, and let me show you our results here. Okay, so this one right here. Some of the highest SAR values in our lab, and these are some of these SAR values are much higher than what you will find there in commercial use. So we clearly, in the lab, in research lab, we can do much better in improving the SAR compared to the commercial instruments out there. So we're trying to communicate with these companies to be able to do this, demonstrate that our particles are much better. So you can see these are really, really high in nanorods. This is the other thing, an important message here is that you can make nanoparticles, but you need to make really high quality nanoparticles, single crystalline nanoparticles. Because remember, you need to have the highest magnetization. And the only way to get that is to make high quality single crystalline nanoparticles. Now that's a bit challenging. So one has to really do that. Okay. For example, if you look at these nanorods, the saturation magnetization is 86 mu per gram. Bulk iron oxide is 92. We're almost there. We can almost get saturation magnetization close to that of the bulk of magnetite. Okay. And this is done through very careful chemistry and fine tuning the synthesis conditions. Another quality check in magnetite, for example, if you are not able to access the low temperature, find out how high resolution TEM, for example, to find out the crystallinity, you can also do another quality check. You can take it to your low temperature magnetic system and look for the Verwey transition. This is a well-known metal insulator transition in magnetite, Fe3O4. It's one of the largest metal insulator transitions known, and it's the first order phase transition, so you see a change in magnetization. Now here's the transition for these nanorods. If your nanoparticles, typically in magnetite nanoparticles, you will not see this at all. It's completely washed away. Only in really high quality particles, you can see the Verwey transition. So again, if you make nanoparticles, magnetite particles, whether they are nanoparticles or nanorods, one thing is to check for the Verwey transition. With that, you can be confident that it's of high quality. I'm almost ready to finish here. I'm going to skip these things. And yeah, we also did some dual functional systems. For example, remember the configuration that I talked about? Here we can make nano clusters. We can actually take silver, gold is even better. You can take gold or silver nanoparticles and use them as a seed, and you can actually have magnetite growing around them, forming this kind of a flower, nano flower structures. Okay. Now these are bifunctional because the gold surface is also exposed, the magnetite surface is also exposed, so you can actually use them for different functionalizations. Okay. And also, hypothermia or heating is not only for magnetic particles, you can also heat up the gold particles. And this is by what is known, if you make 10 nanometer gold particles, you can induce the so-called surface plasmon resonance. And by shining a laser at the right frequency, you can actually cause gold particles to heat up. So you can do the heating both by magnetic hypothermia and photothermal hypothermia. So that's what we did here, and we showed again that indeed by combining the two functionalities of heating, we can do much better than just the magnetic heating itself. So again, these are some examples of different types of hybrid nanostructures that involve interfaces to make this. Now keep in mind that everything that I told you is only half the story, because whatever material we make in the lab, we need to check the biocompatibility. That's the only way we can go to the next step of actually taking it to clinical trials. Okay, so what this means is that you need to study what is known as cytotoxicity. We do these experiments by feeding these particles to cells. In this case, this is an example of a macrophage. We just chose the macrophages because they are very hungry cells. They will eat all the particles, and the cells can go inside the particles, and then you can apply the AC fields to test the functionality. We did a lot of these experiments. This is a slide from a student from Grenoble in France, and it turns out that while I was giving this talk, he said she actually made a video. Oops, let me go back. Let me see if I can play this video. If you can see, it's a video playing. These are glioma cells, and with nanoparticles, and this is a fast frame video. It's a 48-hour video taken over 48 hours that is compressed into a few seconds, and you can see how the cells love to take these particles. So yes, the cells love to eat the particles. Let's feed them and kill them. These are glioma cancer cells. Okay, so clearly the technique works. Hypothermia is a good method, and of course, people ask me, will hypothermia replace chemotherapy? The answer is no, because chemotherapy and radiation therapy are still a very aggressive treatment for cancer. However, as a supplemental therapy, magnetic hypothermia is going to be very effective because it will improve the quality of life of the patient. For example, those of you, if you have friends or family that are taking cancer treatment, that are undergoing chemotherapy, you know the kind of changes they go through. Their immune system breaks down. You can physically see the changes. Okay, so with the presence of with the access to magnetic hypothermia as a supplemental therapy, you can reduce the time between the chemo shots, and you can also decrease the level of the chemo shots, because in the meantime, magnetic hypothermia can be used for controlling it. So I see the hypothermia as a supplemental method that should be available in all hospitals, and should be available soon. Okay. All right. So let me also point out in my last slide, I'm almost done, that in addition to heating, you can also kill the cancer cells by destroying the cell membrane mechanically, magnetomechanically. There are some groups here. So here are some groups that are working in this area. There are other groups also. So essentially, you can actually make synthetic antiferromagnetic discs and then combine that. And what happens here is that when you apply a high frequency field, these mechanically rotate. They're like small motors, and in the process, they destroy the cell membrane. Okay. The advantage of magnetomechanical approach is that the AC fields and the frequencies can be much lower than what we need for hypothermia. You know, I cannot tell you which one is better. I think cancer is really bad. We need all the techniques out there, all new techniques out there. You need to try them out and combine them for effective therapy. The company in Germany, back in 2018, this is 2 years back, they've actually started clinical trials in the US, and which is good news because getting FDA approval is very, very difficult here. It takes a long time. And so this is good news that indeed, hopefully in the near future, there'll be more companies that'll be given the FDA approval for incorporating the magnetic hypothermia into mainstream, so that eventually clinical trials can happen and this can be a real therapy available in hospitals. Okay. Well, this was about a year back, and of course, right now, the whole game has changed now with the advent of coronavirus. So that's where the focus is on right now. So to summarize, to conclude, size does matter. Tamaño importa. I'm not talking about Godzilla, but the other way around, nano size. But nano size has been around for more than 2 decades now. So it's not just the size, it's the shapes, surfaces, and interfaces. Today, hopefully, I gave you some ideas about how to manipulate the surface of nanoparticles, as well as the interfaces, for example, between antiferromagnets and ferromagnets using exchange coupling, and how these can improve the possibility of using these particles for biomedical applications for cancer therapy. I also am an editor for this Journal of Alloys and Compounds. It's considered a premier journal for material science. So I ask all of you to consider submitting papers to us. We have a decent impact factor in this category. Some quick views of some of my students and toys we have in the lab. And finally, on behalf of the Functional Materials Lab at USF, thank you for giving me this opportunity. Thank you for your attention. And like people in Brazil, I've been to Brazil, as I said, 2 times, both times to Rio de Janeiro. Most of you, some of the best research discussions happen in coffee shops and in bars. Thank you very much.
Thank you very much for your talk. So of course here we are online, but like normally there will be a lot of applause. It was really a great panorama overview of everything that is possible. And of course, I know that it's not near the beginning, but there's still a lot of things, a lot of new possibilities with magnetic nanoparticles still for nanomedicine. So people, I will open the session for a question and how it will work for those that are not familiar with Zoom. If you're on the Zoom, just click on participant. It will open the list of everybody, and then there's the raise your hand. So is the way to ask for a question. I already see 2 hands. And for those that doesn't have a microphone to talk, you can also put your question on the chat, and after I will read the question. So let's beginning to Rafael. You can go. Whoops.
Can you hear me?
Yes, please.
Yeah. So I was particularly interested in okay, so I have 2 questions actually, if you allow me. The first one, I was kind of wondering, it might be a stupid question, but how does a particle differentiate which are the cancer cells and which are not? Because I guess that it would heat any particle up, right? If the particle is functionalized.
Yeah. No, first of all, let me say that it's not a stupid question at all. It's very important, right? To be able to target the cells, target the particles to the cancer cells. Now, because I spent all my time on, as a physicist, to explain the particles, the material science, and how we can improve that, there's a whole new other side to it, which is surface functionalization. So this is where you have to integrate exactly the type of target, ligands. There are some ligands which are known to go to the cancer cells. So there's a lot of surface chemistry involved where people have integrated that and have these surfaces functionalized, and eventually you can target those specific cells.
Oh, okay. And will that functionalized surface can affect the magnetization of the particle? Or
that's a very good question. It depends on the quality of the particles. In fact, ideally, if you make super paramagnetic particles, they have to be isolated. The clusters, when they form, sometimes you may not get the best results. So when you do the surface functionalization, the hydrodynamic radius increases, but at the same time, the particles are well separated, and you do have them super paramagnetic. So if you make good quality particles with large magnetization, it should not be a problem.
Oh, okay. No, well, that's very interesting. And the second thing, well, I'm quite interested in, I work with plasmonic antennas into the 2D crystals. So it's more like a nanophotonic area. And I was interested in the part you said that you might get some photothermal and a magnetic hyperthermia from the particles. That's new. And can you talk a little bit more about that?
Yeah, I would refer to that paper that we published a couple of years back. Basically, what happens is that you can actually remember that if you look at the curve, heating curve, you need to get it to that temperature and then hold it steady, right? Saturated in that hypothermia window, which is around 42 degrees Celsius. Now you can use AC field, and this is what my students did. They were kind of playing with it. They used the AC field to heat it up, right? And once you reach the temperature, they turn off the AC field and use a laser. They pulse the laser to keep it constant. So it gives you a second handle to control the heating. So that's the way to put it. But you can use particles themselves to heat it up. But one question that comes to mind is, it's very hard to put fiber optic lasers locally in the tumor area to cause the heating. So that way, AC field has an advantage of immediately producing the heat, and then you can use the plasmonic effects for other purposes.
Okay, thank you very much.
Thank you.
Prabhakaran, you're the next. Yeah, Prabhakaran, are you able to talk? I'm not hearing you. Maybe put your question on the chat because I'm not hearing
yeah, I
anything.
can see the hand raised. No, you can always, I can see.
Now?
Yeah,
I can.
speak just a little louder.
Okay. Professor, thank you very much for the nice, interesting talk on the magnetic hypothermia. And you have shown a lot of results on the anisotropy effects on the heating behavior. And I would like to know that you have shown the exchange coupled nanofarads like FeO and Fe3O4 of different shapes, spheres, cubes, and octopods, right?
Mm-hmm.
And also other shapes. And how their anisotropy affects the heating behavior? Have you quantified the anisotropy through the RF-TDO method?
Yeah, that's a good observation. In all these cases, we also measure which I didn't have time to show, we also measure the transverse susceptibility and get the value for the effective anisotropy. So indeed, the effective anisotropy field that we can find out does increase as you increase the shape anisotropy. So we did correlate between those two.
Okay. 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 Thank you. Thank you very much.
You're welcome.
Perfect. So I don't see any other hand raised, but I have one question, which is always, can you comment a little more about, well, before everybody stopped, about how was the future of really using magnetic nanoparticle with hyperthermia to really cure cancer? So do you think that it will be in the future of 5-10 years? It will be like available for everybody, or can you comment a little more on this?
Yeah, thank you. So as I mentioned, it's not something that is going to replace chemotherapy. That's very clear. That it can be an effective supplemental therapy in addition to chemo, right? So as far as transitioning it to the hospitals, I think the main concern is how do you take these particles out? Once you're done with the therapy, how can the particles be effectively, drained out of the body? And this is something, of course, there's no clear answer to it. It's like a chicken and egg story, right? I mean, to get answers, you need to do clinical trials. And to do clinical trials, of course, you need to get the approval for the process. So we are at the stage where clinical trials are happening. They're happening a lot more in Europe than in the Americas, okay? So we are hoping that the clinical trials will has to perhaps go on for the next 3 to 5 years before it can become a mainstream commercial instrument that's available. It's not a very expensive setup if you look at it. So basically, one has to look at some other issues about how to scale it up. For example, the whole human body should be able to go in there. How to look at these things. But other than that, it's these issues that are related to the toxic nature of nanoparticles and how to get it out of the human body. One thing to keep in mind is that there are natural organs in our human body that their job is to act as screens. For example, your liver, your kidney, they're supposed to collect all the junk anyway. So if the particles end up going to the kidney or the liver, one can perhaps have some kind of a compensation. One can live with that. Certainly the particles should not go to the brain. That's clear. And that's the thing to avoid. So since there are no clear answers of how to limit these things and how to get a good idea, that's the reason why things are getting slowed, I think. I would say that 5 to 10 years is a realistic time frame where you will see this more as a mainstream supplemental therapy available in any of the hospitals.
perfect, but of course it will need a lot of research, to continue some research about on this topic and a lot of topics around. So, well, thank you very much again.
Just one thing, since you just mentioned this, I wanted, this is like something for all the students out there. If you can come up with a biodegradable magnetic material, that's worth the next Nobel Prize. Because it's very hard to find. Most of the magnetic materials that we use here, iron oxide or anything, they're all inorganic. There is no example of a very good, biocompatible biomagnetic material, which has large magnetization. So that would be one of the challenges for the next generation, to come up with ideas where we can have these, then we don't need to worry about flushing it out of the body. And over time, it biodegrades itself. And that's the holy grail here for this community.
So, well, that's a good advice. People, take notes. It's rare that somebody tell you, find this and you will have a Nobel Prize. So now it's just to have it. You don't have to have the idea. The idea is already given. So thank you. So thank you very much for your idea for a Nobel Prize. And thank you very much for your availability to give this talk. It was really interesting. And I hope that at some point you will be able to give more talk. I hope in person at the Physics Institute here.
I look forward to that. Obrigado.
But the Portuguese was good. The Portuguese was good. And thank you everybody also for assisting to this talk. It will be the first of many of other talks. So thank you everybody to be here today with us.
Thank you. Bye-bye. Good evening.